Welcome back.
For something else, isn't it?
Yeah, the queue is
Bij de district mathematics. I hope it's I have to buy there was something from yes.
Okay, so who has solved Leventhal's paradox on the coffee? You don't have that here, right? Nobody? You don't have that. Who solved it? How do we solve Leventhal's paradox?
The only thing you can say is that it's not exhaustive search, because then it happens. So it must be a directed search.
It has to be. There has to be some direction in the search, at least. Yeah. Anything else to help us solve Leventhal's paradox?
You can remove impossible confirmations.
Yeah, but then you would. How do you do that? Because you might only recognize an impossible one when you encounter it. But there must be sort of a, if you think about this there must be a sort of whole areas of impossible confirmations that you just can't get into because the way into that is already impossible. Yeah? So and then you can start imagining that there's sort of a like a valley shape where you can easily move along the valley and you can't go high up the mountains, but you don't need to as long as there's a path because where you like a path through the mountains that goes where you want to go. Yeah.
You should start with the most probable confirmations and go from there.
The most probable. How do you find the most probable confirmation?
So by looking at look at proteins, we see like the beta sheets and alpha units are most presented. So the probability of being in an alpha sheet or
yeah. So that's like a knowledge-based prediction that you do, right? Based on the sequence. Yeah. So we'll actually have a lecture on secondary structure prediction that does roughly exactly that. So you look at a bit of sequence, you look in your databases, and you're like, oh, I see lots of sequences that are similar, and they're all beta sheets. Yeah? But the big problem with that is that a sequence by itself doesn't necessarily have a big preference for being either beta sheet or helical. It depends on the context. It depends on which is the best confirmation combined with the rest of the protein to bury most of its hydrophobic surface, for example. Okay. So yes, and a bit, but yeah. Okay. So so if we think about protein folding, then you can. The simplest way to visualize it is like this, right? So if you have you have a coordinate here, horizontal, which is has something to do with folding. In this case, we can say, well, if we take the root. Did you encounter RMSD already? Or not? Yes, someone mentioned it yesterday. Root mean square deviation. Okay. Basically, you just compare two structures and you calculate the quadratic deviation from between the two, the position of the atoms between the two conformations. Root mean square deviation, and so that's a measure of the distance you are from the native state. So you take the crystal structure as the native state, and you calculate the RMSD, and then you have a folded state at a low RMSD, and you have the unfolded state at a high RMSD. And there will be probably some transition state in between, which may or may not be like the molten globule that I introduced just before the break. So if it was like this, then you don't, you sort of lose Leventhal's paradox, right? Because there's only two states. Yeah? But now it's hidden here because this will, how much variation in structure could you have here? A little, a lot, huge amount in the folded state. How many different structures can you find in the folded state? Not too many. Everybody agrees? It's more than just one, because there can be variations in the structure that are still native-like, right? So you can, like I showed in the beginning of this movie of the fatty acid binding protein, they're all native structures. They all have this beta sheet sort of barrel-like structure, and everything moves a little bit, but the secondary structure and the tertiary structure remain stable. So there's lots of variations here, but then what about here in the unfolded state? Few, many, huge amounts. What is it? 1, 2, lots. Lots, right? The troll counting, if you know your table picture. And then the other thing is that it's not stable. It's not static, right? So a protein might be folded, one protein molecule, and then it unfolds. It goes into some unfolded conformations. Thanks for the battery, by the way, because that was really the thing. Was it? Ah, I should have said it.
You're welcome.
Okay. It is better now, but of course not. It's a demo effect. Right? But it also goes back, right? So it's an equilibrium, and this goes on all the time. Yeah? And in the lecture on structure determination, so experimental methods for structure determination, I'll actually show you some examples of why we know that this actually happens. Because there's data on it. Okay, so something else off. I don't know what it is. Maybe try the other USB port. That's your Something working? Alright. Yes. So it's a dynamic equilibrium. The other thing that it's good to notice is that we have these two states. You can call them folded or unfolded, you can call them A and B. But the probability of being in state A is much larger than the probability of being in state B. How can we see that from this diagram? This has a lower energy, right? Lower free energy. It actually says free energy. We'll come back to that in a minute. Lower free energy means more favorable, means more probable. Okay. We'll get back to this in a minute. Oh, I see I've missed the figure that I wanted to put in. Right. Now, we're going to make it a bit simpler, so I can introduce you some concepts, and then we go back to real protein structure. So, because I want to say something about, I want to quantify this, what we just did sort of in a hand-waving way, about the number of different conformations that you can have in a native state versus the number of conformations in the unfolded state. And something a little bit more quantitative than 1, 2 lots. So, and we go to the so-called HP model. Nowadays people call it the lattice model for protein folding. It was called HP because it has two kinds of particles. And each particle is an amino acid, and they exist in two variations, namely H, which is hydrophobic, and P, which is polar. Not hydrophilic, because then you would have HH, which becomes very confusing. So, and then you have different conformations, and the important thing to count here in this very simple model is the number of favorable contacts, which are hydrophobic contacts. Why? Well, that's because you don't want the hydrophobic stuff to be outside, so whenever two hydrophobic beads make a contact, that's better than having them exposed to the solvent, which is not in this model actually. Sanne will come back to this kind of modeling later on. She's doing this in 3D, and she actually does have an interaction term with the water, which is important, but she'll explain to you why. So, this is already quite old, yeah? 89, that's before I started studying. But if you then, so now we take a very small example. This is a 6-bead molecule, well, let's call it a molecule. It has 4 polar and 2 hydrophobic ones, and these are all the possible unique conformations that you can make of this molecule. All the symmetrical conformations have been filtered out to keep it short. And then there's 2 things that we want to consider here. The enthalpy, which is the energy of the interactions within the molecule, which we count very simply by saying that hydrophobic is favorable, polar is also favorable, but the cross interactions are not. And you can quantify this by a number. The idea. So then you can make all sorts of conformations that have 0 interactions, extended states. And then if you go down, you get more and more interactions. One interaction with all the hydrophobics exposed. This one has the hydrophobics buried. This has 2 interactions, but the hydrophobics are still exposed. And this has 2 interactions, and the hydrophobics are buried for some definition of buried, right? Because they're still exposed on one side, so they make a conflict. So the energy goes from high here, which is unfavorable, to low, which is favorable. And then there's another quantity that we call entropy, which is basically just the number of different conformations that you can have within each level, which goes from very low here to very high there. Right? Who of you have ever played darts? So hitting the bullseye, what makes it difficult? Is that the enthalpy, energy, or is it entropic? Entropic, right? Why? Well, there is no interaction, at least if you're playing dart fairly. You could put a very strong magnet behind the board, then you have an interaction that actually changes the odds, right? But the fact that the reason that the bullseye is so hard to hit is that just simply because there's less of it than the rest of the board. And if you don't play darts a lot and you're not so good at it like I am, then the easiest part to hit is what? The bullseye. The wall. Yes? It's even bigger. It's not because you're aiming at it or because there's some favorable interaction between the dart and the wall. It's just because it's so big, it's easier to hit. Yeah? Okay. So that's what entropy is. It's just the statistics of a bigger thing is easier to hit. Yeah? Elephant hunting is much easier than mice hunting. Yeah. Sometimes amazed by what my brain throws out as it suggests. Okay. Right, so that's entropy. So entropy is the sort of the statistics of molecules. If there's only one state that you need to aim for, statistically it's very hard to get. So you only get it if energetically it makes enough sense. Yeah. And the other way around, so if there's no energy driving you to go into a certain state, it will only happen if there is enough statistical chance of getting there, entropic, right? Okay, so this is the balance that we're talking about. This, by the way, is also the reason why it's so difficult to model. Because you get large numbers here. This is just a simple model, right? But if you have all the interactions in the protein molecule, you have a lot of interactions, there's a lot of energy, right? So you get a huge negative numbers here, and then at the same time there are huge numbers of different conformations possible in each state. So you get large numbers here. And in the end, you're interested in the balance. Yeah, so if you make a few percent error here, and a few percent error there, on the difference between these huge numbers, you might be a factor of 10 or a factor of 100 off. Yeah? If you have a million here and a million there, you make 1% error on each side. The difference between a million and a million may be only 10. You make 1% error on both sides, your difference is 10,000. That's much larger than the difference that you were actually interested in. Yeah, so that's we'll come back to that a little bit more later. That's why in half a century this problem hasn't, one of the reasons this problem hasn't been solved. Okay, now, so this is the statistics. Then there's also the dynamics of this thing. Okay, yes. This is sort of a dynamic picture. This is the same system. It's drawn a little bit different way, but this shows you the possible transitions between these states, because you can't just jump from one conformation to another. I can't instantaneously go from this standing here to standing over there, or I can't move my hand from being up to down instantaneously. It has to go in between, right? It has to move here before it can go there, or I can move in different ways, but my arm can't just be up in 1 millisecond and down in the other millisecond. It's not the way it works. So if you want to go from the completely extended state to the folded state, actually you can do it in 2 steps in this simple model. You can go there, and you can go there. But you can also first go there, then go there, 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 and then there. Right? So that's the dynamics. Now, in the previous slide, I separated states based on energy, but you can also separate them based on characteristics. Well, folded is folded, that's easy. This is sort of extended when there's no internal interactions. And then this is sort of some of these are native-like, a little bit compact. Some of these have interaction. So this could be, if this is the unfolded state, what would this be then? This is the folded state. It could be something like the molten globule state, right? So that looks like that. Well, I mean, not like that exactly, but it's a similar concept. Okay. So now this is the concept of entropy, right? Different states have different number of options to be there, and more options just means it's a lot more likely to get there. Think of a dartboard. Yeah? You get the idea. Okay. And at the same time, it's not like a dartboard because there is actually energy, right? The energy gives you actually. So the dartboard idea is more like the random search. Yeah, it's completely random unless you. But if you know where to hit, if you're good at that, then the protein does that by having these interactions internally that actually help it avoid going into all the unlikely or impossible combinations. You don't have to do all that. If you want to go from here to the city center by a random search, you might end up in Amstelveen and Diemen first, or other places. Right? But if you roughly know which direction you need to go, or if you just follow the gradient of tourists, yeah, then you'll get there much faster. Okay, so we go from a very simple model to a still simple molecule, but a real one. This is a small peptide. If you look carefully, it's a little bit different than regular amino acids because it has a second carbon in the backbone. That's not so important, but in case you're wondering whether this is an error, drew it wrong, no, this is actually how to make them out. And this is again a video of a simulation. And I'll turn off the light. And so you can see this is actually much more flexible and dynamic than the protein that I showed you previously. There's 2 reasons for this. One is because it's such a small molecule, it's much less stable than the protein. Yeah? The other is that there's actually 3 reasons. the time scale of the simulation is much longer. This is 200 nanoseconds. The other one was 5. This one doesn't have a counter on the screen because I made. Well, I didn't make this video. But the guy who made this video didn't put the counter, put the time on it. But the whole thing, it's in its second loop already. But the whole thing is about 200 nanoseconds. The third thing is that such a small peptide can sample a lot more of its conformational space in 200 nanoseconds than a protein can, because it has much more conformations to go through. Right? So that all that together ends up being much more fast. So this is actually the helical state, which was measured by NMR on this molecule. That's supposed to be the native state. This is a Tremper molecule, but it's actually it's real, but it's not. It doesn't have a biological origin. It doesn't have a biological function. It's just to investigate the physics of it. By the way, we have a later lecture on explaining these molecular simulations that I now show you the results. So you can plot this in a different way by plotting the root mean square deviation versus this helical native structure as a function of time. And then you can see it starts low, then it goes up, and then it goes down again, and it goes up again, and it goes down. So you see lots of transitions from the folded state, which is, let's say, below 0.1 nanometers, and the unfolded state, which is anything more distant from the native state. And of course, if you then look at each time it goes to a native state, they're not identical, but they're very similar. Yeah? You can see the similarity because it has this helical core. But you see there's lots of small scale variations there as well. On the other hand, if you take like 4 random snapshots from one of the unfolded, one of the moments that it's unfolded, different moments that it's unfolded, you see very different states. Yeah? This is completely open, this is sort of like a beta hairpin, this is somewhere in between, this is almost completely extended. So they're all roughly the same. They're not the same, but how different are they? How many are they? They actually did a clustering of this. So they used the same criterion of 0.1 nanometer as they used to define the folded state. So now we have a folded state that's anything within 0.1 nanometer RMSD from the native conformation, and all the other states are similar distance clusters from your folded state. And then they ended up with about 1,000 clusters. So that's an interesting number. So let's examine what that means. Because this is only so we have this is a 7 residue peptide. So we only have why does it say 21? Oh yes, because we say 3 states per residue, right? So then we have 21. Oh, sorry. Yes, no, it's because it has this extra carbon in the backbone. It has an extra angle. So it has 3 angles per residue. So it has 21 angles in the backbone. So 3 to the power of 21, that's about 10 billion possibilities. That's not a huge number compared to what we saw for the protein before, right? But it's a small peptide. Yeah, that's a big difference. But even out of this 10 billion, we actually effectively see only about 1,000. Yeah? So that's our way out of Leventhal's paradox. Yeah? Then you can still, and we're not going to do that in this course because that's really still quite difficult to examine exactly what the properties are of these 1,000 clusters that one of them is the folded state and the other 999 are the unfolded states, if you will. What makes them so special? Why are these ones sampled and none of the other 10 billion, right? Okay, but then if we go, so we have now, let's say we have 10, the equivalent of this peptide I just showed you in normal amino acids would be a peptide of 10, which would normally fold in about 10 nanoseconds. 10 to the minus, nanosecond is 10 to the minus 9, it's 10 to the minus 8 is 10 nanoseconds. And we have about 1 billion possible structures. But we know from the simulation now that we only sample, the molecule only needs to sample about 1,000 of them to be able to fold reliably and reversibly. Right? It's like what Alphensen did in his experiments with refolding, but now it's done in simulation. Right? You don't just watch till it goes close to the native structure, but you see it going back and forth all the time. That's dynamic equilibrium. So in 10 to the minus 8 nanoseconds, sorry, in 10 to the minus 8 seconds, we can do about, the protein does about 1,000 conformations. So, so we can make a difference between the number of possible structures and the number of apparently relevant structures that allow you to fold. Then if you extrapolate this to a protein of about 100, still a small protein, with about 100 residues, we know from experiment these fold in about 10 milliseconds. They have about 10 to the 19, that's the number I had before, right? Different conformations. But if you then take this ratio, so it takes 10 nanoseconds to sample 1,000, so it takes, so how many do you need to sample? How much can you sample in 10 milliseconds? That's a million times this, right? So that's billion. So to fold 100, for 100 residue protein to fold, they will need to sample something on the order of 1 billion different conformations. That's much less than 90. And if it can do one of these in a picosecond, then we've solved Levendal's paradox here, right? So somehow, because of these interactions that happen within the molecule, the proteins are able to sample only a very, very tiny fraction of all possible conformations, avoiding all the impossible ones, so to say, or irrelevant ones. They're not impossible, they're just irrelevant. Because some of them are impossible, but most of them are just irrelevant. Something like that, right? So, but then there's still a big step from here to understanding what the mechanism is behind this folding in detail. Right? And that's, as I already said, that's too much detail for this course. We won't be able to get into all that. So, and in simulations, we've now solved this for peptides. Right? So then maybe for proteins we can also solve it in simulations. But we'll have some example of that later. That's also one of the papers for the exam. But I'll show you actually that it's not really solved for proteins. To be honest, it's not really solved for peptides either, because this is the simulations I showed you. This is work from 1999. That's a while ago. And they of course try different peptides, and it doesn't work for all peptides. So the model there is just not good enough, and they've been of course improving it ever since. And it's still not good enough. It's very hard to get the model right so that it works for all sorts of peptides. If it doesn't work for all sorts of peptides, it's very unlikely to work for proteins. So it's still not there yet. Okay. The other thing is, what happens if you change the temperature? What do you expect? Let's go back to it. Sorry? So when should it unfold? When it goes up, it should unfold, right? That's what you expect. Okay. That's actually what you see as well. So this is still from simulations. This is still work from around 2000. So the simulation that I showed you before was at 340 Kelvin. It looks different, but that's because this was done at a smaller time scale. So this is only the first quarter of the whole simulation. This was still very expensive simulation work in those days. So when they were doing different temperatures, they made the simulation a bit short. And then everything below 0.1 we call folded, like before, and everything above them is unfolded. And then you see that if you go, this is only 40 kelvins lower, yeah, then most of the time it's folded. It unfolds only rarely and very briefly. This is too short simulation, right? So this is folded more often than this one. Okay, so, but you also see that you only have a few events where it unfolds. So apparently in the simulation it happened to stay unfolded a little bit longer than you would expect an average. So to be able to get really good statistics, you want to see the folding unfolding tens of times, preferably 100 times. Otherwise you don't get real good statistics. If I throw at a dartboard 2 times, you can't tell how good I am. Well, even if I hit the bullseye twice, it could still be luck. But if I do that 100 times, then it's not luck anymore. At least it's very unlikely. You can calculate a p-value for that. Yeah? Okay. So this is too short to really get good estimates of the statistics there. That's really also a problem. But still. Yeah, you see here now the fraction of folded starts to drop even further, and even just 20 degrees higher, you can see that the fraction of folded has really dropped by quite a bit. You can actually make a plot out of this, and it looks a little bit like this. So you have almost completely folded at about 300 kelvins, and it goes to less than 30% folded at 360 kelvins. This is, as for biology, this is a normal temperature range. As physics goes, this is quite a narrow temperature range. Yeah, it's 300 Kelvin and then 20% up. Yeah, you could go from 100 Kelvin to 3,000, or from 10 to 3 million. Yes, but still, I mean, so what I'm trying to say is that you don't have to change a lot about the physical conditions for this protein molecule, this peptide actually, to respond to that. Right? So it's quite sensitive. Okay, so so why does this happen? Why? Because it makes sense. Nobody's surprised that this happens. Why does it make sense? Why does it make sense that, well, yeah, of course, it unfolds when you raise the temperature. Everybody who's boiled an egg knows that proteins respond when you, actually, you know, the protein, the Dutch word for protein is eiwit, which is egg white, because it's for a protein. Why is this not surprising?
Because the water molecules will start moving at a more rapid speed, which allows the protein to unfold.
Well, all atoms move fast.
Yeah. Everything moves faster, also the protein itself. Yeah.
Compared to the folded state, the unfolded state becomes more favorable, entropy-wise.
Yes.
Or like if you calculate the effect of the entropy becomes stronger with high temperature.
Yes, so that's the right direction. Yeah. So the start of your answer was rephrasing the question in terms of energy and stability. So it's the balance. So you remember the previous slide. Where is it? This one. There's a balance between favorable enthalpy, which you have when you have lots of interactions, and favorable entropy, which you have when you have lots of different conformations. And this balance actually depends on the temperature. Yeah? So low energy means favorable. But what is this with entropy? Right? So actually entropy is sort of like an energy, but it's not quite that. It actually is an energy in a scale with temperature. I'll have a formula in a minute. But that means that the energy that is represented by the entropy becomes bigger when you're at higher temperature. Yeah? Right. So if you know that, then it makes sense that if you raise the temperature, then this factor which drives the conformation, sorry, the state with lots of different conformations to be more favorable, because that's the entropy, and that's the unfolded state, right? And if you go to low temperature, then it's the enthalpy that wins. So yes, skip a few slides. So now we can go here. So if you think about I have to make a step. You remember this one, right? So here you have different states. So there are different ways to represent these states. One of them is to think of the different conformations to be projected onto a map. Could be a two-dimensional map, like the Ramachandran plot, but in reality, it's a multi-dimensional map. But it's very hard to draw a multi-dimensional map. So usually we draw a two-dimensional map, and then the energies go up, right? This is some representation of different conformations. There's different ways to do that. Now you can, if we make it simpler and we go for one-dimensional again, then you have you can have a state where you have few different conformations because it's only a narrow region in my coordinate space, and it has a very low energy. Yeah, it makes it favorable. But this one actually has a much less favorable energy, but it's much wider, this state. So there's many more different conformations. And the formula that goes with this is that the difference in free energy between these two states, that's the delta G, is first of all the difference in energy, which is this depth, but then minus the difference in entropy, which is in here like the width of the state. So entropy here is smaller, entropy there is larger, times the temperature. Right? So there you get this temperature dependence. And actually I want to show something. I'm missing something. What am I missing? No. So does this make sense? Yeah? Ah, good. So now the final example I want to show you is of a small protein. It's about, what is it, 35 residues or so. It's a very small protein, where in about 2010 they did some extensive simulations. This will reappear in the later lecture, but I just want to end with it now because it's a nice example. And they basically did exactly the same as what they did with the peptide. And one of these simulations was started in a completely extended state, and the other one was started in the crystal structure state, and you really can't see the difference. And also the changes, I think it's this one that started at the crystal structure state, and it just immediately unfolded. And 10 years before, they would have thrown the simulation away because they would never come back. But they just kept on simulating. Look at the time scale, it's microseconds. These guys are crazy. This guy made his Shaw, Bernard Shaw, David Shaw, sorry, Bernard Shaw is not there. He made his fortune on the stock market and then founded his own research company. They have like hundreds of millions worth of special purpose built computers. You never guess what they called how he called his first computer. Anton. We never met, so it's not happening. Anyway, so he could afford, and he's still one of the biggest labs in that sense. He could afford doing microsecond simulations, I mean hundreds of microseconds. This is close to milliseconds, right? Now, the big problem is that you see it unfold, fold, unfold, only like a handful of times. So this is still way too short for a 35 residue peptide, the protein, right? But it still shows that it actually folds spontaneously. So the model is a model. It's not correct by definition because it's a model, but at least it gets something right. Yeah? And it's two different simulations. They're just here stacked on top of each other to make the figure a bit smaller. Two independent simulations. Okay, now, what can you do for this? You said something about when you want to predict the structure of a protein, look for the most likely conformation. You can do that from a database, but you can also do that in the simulation, right? So you have two states here, basically. You have the folded state, and you have the unfolded state. And you can collect, like I did with the peptide, you can collect the probability of the two states, and that says something about the relative stability. The more stable state should be the more probable one, or the other way around, the more probable one should be the more stable one. So which measure do we have to express stability? Probability is easy. So it's just counting along the axis how often you see one state or the other. How do we express stability? It's been mentioned a few times already. It's free energy, right? We'll come back to free energy in lecture on thermodynamics, but I just want to get the concept sort of slowly growing in your mind. I'm going to skip this one for now. So they actually do it this way, right? So they introduce something they call a reaction coordinate, which is like the RMSD, except a little bit smarter. And the reason for that I don't want to go into. And then this is like a free energy diagram that I've shown before on the board, except they turn it around. So the free energy now is horizontal, and the reaction coordinate is vertical, so that it aligns with the vertical RMSD axis. Right? So this is folded, below RMSD, close to the native state. Below reaction coordinate means the same, it's folded. And there you have the unfolded state. Yeah? And then there's a transition state in between, and you can see that this nicely corresponds to this coordinate. Right? So this is the folded state, this is the unfolded state, and then there's a state in between where it just sort of zips past, right? So it has to go through some intermediate states to go from folded to unfolded and back, but it's not a particularly favorable state because it doesn't stay there a lot. So you see that back in the free energy diagram. Now you can express this same in a formula, and don't worry, we'll come back to this, but I just want to, as I said, let this start growing in your mind. So you can relate, so the free energy is over here. This, I don't remember the pronunciation of this Greek letter. It's a xi. Okay, so xi is the reaction coordinate, right? So you have a probability that's a function of the reaction coordinate of xi, and you have the free energy that's a function of the reaction coordinate. And they are related by this, right? So if the free energy goes down, then the probability goes exponentially up. Yeah? You also see the temperature in there. So it depends on the temperature. And you can reverse this as well, where if you know the probability, which we actually know because we can derive it from the simulations to some measure of accuracy, you can just count the probability of the different states. You take the logarithm of it times t times Boltzmann's constant, and you get the free energy. Yeah? So there's two ways to get the free energy. This is the statistical way, and this is the thermodynamical way, where you have the interaction energies and the entropy which is a measure of the number of states, number of conformations in each state, and you also get the free energy. Yeah? So, and we're using different symbols for the different terms, sort of. It looks maybe random. There's reasons for that, but that's in chapter. That's explained in chapter 13 on thermodynamics. Okay. Main points. Proteins are flexible, they're always moving. It's because there's temperature. The crystal structure is only an average. It's still very useful, but it's only an average. We can actually do this in a simulation. And I think I've now explained to you how we can get around Leventhal's paradox. Are there any questions before we go to the practice? No? Okay, see you in the... Yeah? Yeah? I'll get a bit closer to you. Yeah.
Which one is it? Because there are two movies at school. They have the same one that they're on the Alex.
It's not logical why it's called.
Oh ja. Ja. Dank je, Janne.
Yeah, they don't live in the room.